There is a part of your brain that is physically shrinking, not because of aging, not because of disease, because of the way you live. The hippocampus is the region of your brain responsible for memory, emotional regulation, and your ability to learn. And decades of peer-reviewed research now confirm what neuroscientists have known for years and almost no one is talking about in plain language. Chronic stress makes it shrink. This episode is not about feeling stressed. Everyone feels stressed. This is about what is happening at the biological level when stress becomes your baseline. In your hippocampus, in your genetic code, in the nervous system of the children you may one day have. This is the Brain Care Podcast, Season 2. I am the doctor. Your body was built for emergencies. When a threat appears, your hypothalamus fires a signal. That signal reaches your pituitary gland, which tells your adrenal glands to release cortisol and adrenaline. Your heart accelerates. Blood shifts away from digestion and toward muscle. Your prefrontal cortex, the part responsible for rational thinking and decision-making, goes offline. And your amygdala, your ancient threat detection center, takes command. This is the fight-or-flight response. It was designed to keep you alive for about 15 minutes. It was not designed for a Tuesday morning of unread emails, financial pressure, social comparison, and a global newsfeed that delivers 27 crisis events before you have your first coffee. When cortisol stays elevated not for minutes, but for months. A systematic review published in 2025, synthesizing 25 peer-reviewed studies, confirmed what the field has been building toward for decades. Chronic stress reduces hippocampal volume. It impairs synaptic plasticity. It produces measurable deficits in memory performance. And the primary driver is glucocorticoid-induced neurotoxicity. Cortisol, at sustained levels, becomes a destructive force inside the most important structure in your emotional brain. Your stress is not in your head. It is in your hippocampus. Research published in PMC by the National Institutes of Health confirms this directly. Prolonged elevation of cortisol impairs synaptic connections, suppresses the formation of new neurons, and reduces hippocampal volume. These are not theoretical risks. They are measured changes in tissue. And there is more. The blood-brain barrier is a highly selective membrane that separates your bloodstream from your brain tissue. Under normal conditions, it blocks most inflammatory molecules from reaching your neurons. Under chronic stress, it becomes permeable. A 2024 review published in Frontiers in Molecular Neuroscience confirmed that chronic stress alters the integrity of both the gut barrier and the blood-brain barrier, allowing pro-inflammatory compounds to infiltrate brain tissue directly. A 2023 systematic review in Frontiers in Psychiatry found that neuroinflammation is present in up to 27% of patients with major depressive disorder and is associated with a more severe, chronic, and treatment-resistant trajectory. The HPA axis, when dysregulated by chronic stress, also disrupts the prefrontal cortex and limbic structures, contributing to exactly the depressive symptoms that modern psychiatry struggles to treat. Research published in PMC in 2025 traces this entire cascade and maps it directly to the same hippocampal deterioration that neuroscientists have been documenting for 30 years. You are not anxious because your life is hard. You are anxious because your neurobiology has been reconfigured by sustained cortisol exposure. And these are two very different problems with very different solutions. Here is what almost no one tells you when they talk about stress. Chronic stress does not only change how your brain functions, it changes how your DNA is read. This is the science of epigenetics. Epigenetics is not about changing the sequence of your DNA. It is about changing which parts of your DNA are turned on or off. And chronic stress is one of the most powerful epigenetic forces known to biology. The NR3C1 gene codes for glucocorticoid receptors in the brain. These receptors are the biological mechanism that tells your HPA axis to stand down once the stress has passed. Think of them as the off switch. Research published in peer-reviewed journals has consistently shown that chronic stress causes hypermethylation of the NR3C1 gene. When this gene is overmethylated, it is silenced. The off switch stops working. Your brain loses its ability to downregulate its own cortisol response. Stress teaches your brain to stay stressed. BDNF is brain-derived neurotrophic factor. It is the primary protein responsible for neuronal growth, synaptic maintenance, and neuroplasticity. Without it, your brain cannot repair itself or form new connections. A 2025 study published in PMC found that chronic stress was directly associated with measurable methylation of the BDNF gene. Exercise increases BDNF. Chronic stress suppresses it, and when BDNF is suppressed by epigenetic silencing, the effects are not transient. They persist even after the stressor has been removed. And now the dimension that changes everything. Research using cross-generational models demonstrated that early life stress transmitted epigenetic changes to the next generation. Human cohort studies are reaching the same conclusion. Studies on survivors of the Rwandan genocide and Holocaust survivors have documented similar transgenerational cortisol dysregulation. Your unresolved stress is biological information passed to the next generation. You are not only carrying your stress, you may be transmitting it. File number 002, subject James Fallon. In October 2005, neuroscientist James Fallon was in his lab at the University of California, Irvine. He had spent nearly 2 decades studying the brains of psychopaths and violent offenders. His research was standard neuroimaging work, PET scans comparing brain activity in murderers, schizophrenics, and healthy individuals. At the bottom of a stack of scans, he found a brain that looked obviously pathological. Low activity in the orbital cortex, low activity in the anterior cingulate cortex, exactly the pattern he had documented across decades of research in violent individuals. He assumed it was a processing error. He checked his equipment. It was working perfectly. He broke the research blinding to identify the scan. The brain in the scan was his own. Fallon then underwent full genetic analysis. The results confirmed high-risk alleles for violence-associated genes, including the MAOA gene variant. He carried the neurological profile and the genetic profile of a psychopath. He then traced his family history. One of his direct great-grandfathers, Thomas Cornell, was hanged in 1667 for murdering his own mother. That branch of the family produced 7 other alleged murderers, including Lizzie Borden. James Fallon became a neuroscientist, a husband, a father, and a professor at one of the most respected research institutions in the world. He is not a psychopath in any clinical behavioral sense, despite carrying the full profile of one. His explanation for this is precise, and it is directly relevant to everything this episode has investigated. Epigenetics, the key developmental window, when the HPA axis and NR3C1 methylation patterns are being calibrated by early life experience, was characterized by relational safety and secure attachment. The early nurturing switched the trajectory. The same high-risk alleles that predispose to violence, when combined with a safe and nurturing early environment, can become resilience genes, resistance genes. The genetic code does not determine the outcome. The early epigenetic environment does. This is the case that reframes the entire conversation. Your stress is not a character flaw. Your epigenome is not fixed. The genes that create vulnerability are the same genes that create resilience under conditions of biological safety. Fallon's case was reported in Smithsonian Magazine, NPR, The Atlantic, and BBC. His book, The Psychopath Inside, published in 2013, documents the epigenetic analysis. His TED Talk has been viewed millions of times. The biology is documented, the lesson is available, the application is yours. The same epigenetic plasticity that makes your brain vulnerable to chronic stress makes it capable of measurable recovery. Let me be exact about this, because this is not motivational language. This is peer-reviewed biology. BDNF, the neuroplasticity protein suppressed by chronic stress, is significantly upregulated by aerobic exercise. A 2024 systematic review in PMC found that BDNF increases measurably following high-intensity aerobic exercise. These are the biological conditions for neuroplasticity restoration. Controlled breathing, specifically slow diaphragmatic breathing at approximately 6 cycles per minute, activates the vagus nerve. And vagus nerve activation is one of the most direct inputs you have into downregulating the HPA axis. A 2025 study found that transcutaneous vagus nerve stimulation directly inhibits cortisol release during mental stress. Additional research confirms that slower breathing patterns increase heart rate variability. The gut-brain axis is the bidirectional communication network between your enteric nervous system and your central nervous system. A dysbiotic gut microbiome increases intestinal permeability. This allows pro-inflammatory bacterial compounds into systemic circulation. Those compounds compromise the blood-brain barrier, and the inflammatory cascade that follows exacerbates the neuroinflammation already triggered by cortisol dysregulation. A 2024 review in Frontiers in Molecular Neuroscience confirmed this mechanism directly. Dysfunctional gut-brain communication is repeatedly linked to anxiety and depressive behaviors. Gut dysbiosis perpetuates and amplifies HPA axis dysregulation. This is not wellness content. This is applied neurobiology. Your genes are not your sentence. They are your starting point. What has been missing is a framework that integrates them into a coherent, personalized biological protocol, one that accounts for your constitutional stress profile, your epigenetic baseline, and your ancestral patterns. Your stress is not personal weakness. It is a biological signal operating inside a system that was designed to amplify it, not resolve it. And the science tells us something specific. The earlier you understand your own constitutional stress profile, your dominant HPA axis pattern, your epigenetic vulnerabilities, and your genetic tendencies, the more precisely you can apply the interventions that are documented to work. This is what I built the neuroenergetic blueprint of the 5 elements for. It maps your constitutional stress profile using your elemental typology, your ancestral patterns, and your personal biological tendencies. It identifies your dominant stress response pattern and gives you a personalized starting protocol for HPA axis restoration. It bridges epigenetic science with the 5 elements framework in a way that is specific to you, not generic to everyone. The link is in the description. For those ready for full biological recalibration, the Elemental Performance Lab is a 12-week physician-led program. Cortisol mapping, BDNF restoration protocols, gut-brain axis repair, personalized nutritional intervention, supervised by a medical doctor. The link is in the description. Next episode, the polluted brain, fluoride, microplastics, and heavy metals. What they are doing inside your neurons right now, and who decided that was acceptable. This is the Brain Care Podcast, Season 2. I am the doctor. Protect your brain, because no one else will.